Hello everyone, today we are talking about bacteria. Now, if you recall from our taxonomy lessons, bacteria can be broken down into two different kingdoms, eubacteria and archaebacteria. So we have a few goals for us today. The first is to be able to describe unifying anatomy or morphology of bacteria. So there's a few characteristics that make them similar. Explain the importance of bacteria, to describe the types of reproduction, and to describe some of the bacterial habitats can be unique to their kingdoms. I hope that you recall the term prokaryotic because bacteria are prokaryotic cells. They are unicellular, so there's only one of them, one cell. There's no nuclear membrane around the genetic material, and you can see that here. And there's no other membrane-bound organelles like chloroplasts or mitochondria. They're small, usually less than 2 micrometers, and there's one circular chromosome, and so that circular chromosome is wrapped up in there. So bacteria are prokaryotes. They are the smallest known living cells. Notice I said living. And all are unicellular. So all bacteria are prokaryotes and unicellular. They're also found everywhere. And when I say everywhere, I mean literally everywhere, on probably every surface. I have a few images here. This is the head of a pin. And all these little orange things, those are bacteria. And this is your teeth, and so this is bacteria on plaque on your teeth. There are over 80 species of bacteria in your mouth alone. Now, bacteria are really important when we talk about evolution because bacteria are thought to be some of the first forms of life, meaning one of the first cells. But due to insufficient fossil records, it is challenging for scientists to figure out what bacteria evolved from. We've talked about LUCA before, last universal common ancestor of all organism, and scientists believe that LUCA was likely to have been a small single-celled organism not too dissimilar from bacteria, from modern-day bacteria. Though some scientists have discussed that it's possible that its genetic material would have been a simpler, more rudimentary structure. We've talked about how bacteria are everywhere, and it's important to know that some bacteria are considered to be bad. Often when you think of bacteria, you think of germs. But the reality is that bacteria are also very important and are beneficial. So here's just a few examples. Bacteria help us produce cheese, yogurt, and there's also bacteria in our gut that helps us produce vitamin K, which is essential in our body. However, when bacteria are bad, they cause disease, we call them pathogens. Next up on what we have to talk about are the structures of a bacteria cell. The first structure is the capsule. As you can see, you follow these arrows down. The capsule is that yellow outer coat that helps keep the cell from drying out and also helps it to stick to other cells or substrates. So that's the capsule. Now, just inside that capsule, we have our cell wall. Now, cell wall should be a structure that you're familiar with. Of course, plants have a cell wall. And most students by this point know that animal cells do not have a cell wall. Well, bacteria cells also have a cell wall. There are some differences between the bacterial cell wall and the cell wall of plants or fungi. For example, bacteria cell walls contain peptidoglycan. That's that word down here, peptidoglycan. if you break that word up, we get peptide, which is peptides, so amino acids, which are the building blocks of proteins, and glycan, which is a generic word for saccharides or sugars. So peptidoglycan is composed of proteins and sugar. If you compare the cell wall of bacteria to plants, plants' cell wall are composed of cellulose. Fungi, it's composed of chitin. Now, given that we're talking about bacteria, we are talking about two different kingdoms of bacteria. We're talking about eubacteria and archaebacteria. It's important to note that archaebacteria usually lack that peptidoglycan composition of the cell wall, but there's always exceptions, and so methanobacteria, a type of archaebacteria, does have peptidoglycan. So there's definitely some variety within the structure of the cell wall, even within bacteria cells. Anyhow, point is, the cell walls are different. The purpose of the cell wall is fairly similar. It's a thick outer covering that helps to maintain the shape. Ribosomes is another structure that you should be familiar with. Cells have ribosomes, so do bacteria cells. Ribosomes are what create proteins. Well, they have a big part in creating proteins. And when we're looking at bacteria in electron micrographs, it gives bacteria this granular appearance. So that's what you're actually seeing, the ribosomes. And those are all those little dots inside the cell. Our next structure is called the nucleoid. And so this big blue line is pointing towards the genetic material. Now, there is no nucleus in a bacteria cell because remember, it's a prokaryote, so there's no nucleus. What they have instead is a ring made up of DNA that we call the nucleoid. Not a nucleus, a nucleoid. Our next structure is a really interesting one. It's called the flagellum if there's one, or flagella if there's more than one. Now the flagella is like a tail that comes out of the bacteria and it whips around in circles in order to create locomotion or movement. And so it will propel the bacteria in some direction. One thing that's I find pretty interesting about the prokaryotic flagellum is that it runs off of sort of a motor process right at the base of the flagellum. And so what happens is the motor causes spinning at the base, and as a result, this causes the rest of the flagella to sort of flail around behind it. But all of the power in that movement is coming from that base. I've included a few videos here that just show you what it looks like when it's moving. Definitely give it a watch. It's interesting to see. The other important thing is there can be one or more flagella on a bacteria. Next up, we have the cytoplasm. Just like in our happy little eukaryotic cells, prokaryotic cells have cytoplasm, which is just that clear jelly-like substance that suspends the organelles and takes up most of the space in the cell. Okay, last one, the pili. The pili are these hair-like structures around the sort of embedded in the capsule of the bacteria, and these pili help the bacteria stick to other things, whether it's a tissue, other bacteria, surfaces, whatever. They're often found in bacteria that are pathogenic, so disease-causing. Now, later we will talk about cilia, and so pili are not to be confused with cilia. The purpose is totally different. When classifying bacteria, one really important thing we use is whether they are known as gram-positive or gram-negative. What this means is we can stain the bacteria with a special stain, and if it turns purple, it tells us that there's peptidoglycan in the cell wall. And so what that'll look like is it'll turn purple because of that stain. If it's gram-negative, then it will stain pink. And so gram-negative bacteria have a thin protein layer on their cell wall and no peptidoglycan, so they're pink. And they're generally a larger and more diverse group of bacteria, whereas our gram-positive have that thick protein layer, that peptidoglycan, and so they turn purple. There are 3 basic shapes for bacteria. I mean, there are actually a few more, including Vibrio, which is like a bean shape, and Spirochaetes, like a coil, but let's focus on 3. So the first is spherical, or coccus. So that's these guys over here. They look like a sphere, and we call them coccus. The next is a rod-shaped. So these look kind of like a rod, or like a breadstick. And so we call them bacillus. And our last basic shape is coiled. We call it spirillum, and they have these sort of spiral type shapes. Now, bacteria aren't always going to be by themselves. Sometimes they form colonies. And so we have a few names when we're talking about their colonies. Mono means a single cell. Diplo means a double cell. So if we have 2 coccus-shaped spherical bacteria, we would call it diplococcus. So there's 2 forming a little diplococcus colony right there. We also have strepto, and so that's when bacteria form chains. Here we have streptobacillus, because remember, these are our rod-shaped bacteria. And finally, we can have staphylo, which is clusters. And sometimes they're random or disorderly. Sometimes they look more like grapes on a vine. And so these are staphylococcus. The way that these bacteria form colonies also helps us to name and identify them. So we've talked about shapes, we've talked about gram-positive or negative. The next major topic we need to talk about is reproduction. Bacteria reproduce through a number of different ways. Their primary method of reproduction is through binary fission. Binary fission is comparable to mitosis in the sense of we have one cell that duplicates its DNA, the cell divides, and then we get 2 identical daughter cells. Now the difference, of course, is that mitosis involves a nucleus. So mitosis describes the division of the nucleus. Binary fission doesn't happen with a nucleus. So this is a form of asexual reproduction. What you have here is an image of fission in action, so binary fission. So we started with 2 cells, and then each of them duplicates and reproduces again, and reproduces again, and reproduces again. And so what we end up with is thousands of bacteria in a very short amount of time. In ideal conditions, which usually means warm and moist, some species can divide every 10 minutes. That's very fast. So, you know the whole 3-second rule? Maybe not if you're worried about bacteria. So then the question is, what stops them from growing? What stops them from dividing? Why don't you pause the video and see if you can figure it out? Hopefully, you pause the video. The major impact, or the major thing that stops them is actually the resources available to them, whether that be food, available food sources, or space. as well as waste. If there's not enough space to get rid of their waste, then that can build up, and well, waste is poisonous to any organism. That's kind of why it's waste. The next type of reproduction is conjugation. Conjugation is sort of as sexual as it gets, at least for bacteria, that is. Because in the case of conjugation, we need two bacterial cells that join together, and what's going to happen is they're going to exchange their genetic material. So here's a wee bit of genetic material, it's going to get passed over to this bacteria, and now we have both bacteria with that genetic material. Earlier, I talked about those pili. Oops, that's a pen. I talked about those pili, and so there's actually something called the conjugation pilus, which helps those two bacteria cells physically connect to each other, create that bridge so that the genetic material can be passed from one to the other. Here's the thing. Just like with shapes of bacteria, there's also tons of different types of reproduction. We talked about binary fission and conjugation, but there's also something called an endospore, which is where the bacteria create dormant cells that can stay dormant for years and years and years, which are very resistant to environmental stress. There's also something called transformation, which is where bacteria take in DNA from the environment and incorporates it into its own genetic information. So it's basically bacteria transforming itself. And there's also something called horizontal gene transfer, where DNA is passed from one species to another. So it's similar to transformation, except instead of bacteria just finding DNA, it's passing from one species to another. Bacteria are very little. Not as small as viruses, but they're often very little. And there's a few reasons for this. By being little, they have a high surface area to volume ratio. So their surface area is high compared to their volume. This is important because it allows them to diffuse nutrients, secrete wastes, and it's much faster when you're little because of that surface area to volume ratio. Picture this. We have a bacteria that is just 1 cubic units worth of bigness. Doesn't really matter what the unit is. And so its surface area is going to be 1 by 1 times 6. So 1 by 1 times 6 is 6 for surface area. And 1 times 1 times 1, so length, width, and height, is 1 for volume. But if we consider a 2 by 2 cube, then the size has gotten bigger, and we can calculate the surface area again. So we have 2 by 2, and there's 6 sides, so that would give us 24. And if we calculate the volume, it's 2 by 2 by 2, length, width, and height, and so that gives us 8. We can then convert this right here, change it to a ratio, so it's 24 by 8, and if we simplify that, it would become 3 by 2. So this right here is a much less favorable ratio than our 6:1 ratio. So the smaller the organism, just based on that surface area to volume ratio, the better it is going to be able to diffuse substances across its membrane. So things like bacteria are small for a reason. What I want you to consider is humans. Humans are large, very large compared to bacteria. What is it that humans do that allow us to overcome that surface area to volume ratio? Because our surface area to volume ratio is not very good. What do we do? Pause the video. Can you figure it out? So I hope you paused the video and gave yourself some time to think about that. Now, in grade 10, you would have learned about tissues, specialized tissues and systems in the body that sort of create a division of labor. This division of labor has allowed larger organisms to overcome these limitations of diffusion and that surface area to volume ratio problem. So for example, one requirement of cells is to obtain nutrients. An example of that could be glucose. So all of my cells need access to glucose. Glucose is transported around the body via our circulatory system. Okay, next up we have classification of bacteria. We have discussed this, that there are two types of bacteria. We have archaea over here, and we have eubacteria over here. And there are some differences, which makes sense because they're in two different kingdoms. When classifying them, we look at a few different things, including origin, shape, and arrangement. We already talked about shape and arrangement. So we have coccus, bacillus, or spirilla for shape, and arrangement could be by itself, diplo, strepto, etc. Now their origins are either archaea, domain archaea, or domain bacteria. So here are some major differences. Over on the archaea side, we have our ancients. Archaea means ancient. They're the oldest organisms on Earth, the oldest organisms. And they live in very extreme environments. So we call them extremophiles, extreme lovers. Their cellular biology, genes, and DNA are quite different from eubacteria. If we come over here and look at eubacteria, eu means true. When you think of bacteria, you're usually thinking of eubacteria. Now, eubacteria are the ones that are found in tons of environments. That's kind of why you're usually thinking of eubacteria when you think of bacteria. They are ones that are more commonly encountered, and that sort of lends itself to why they are considered more successful than archaea, because there's just so many of them in so many places all over the world. Now, if you recall, we have a term called autotroph, which is where they make their own energy. They use solar energy to so-called fix carbon dioxide. An example of that is cyanobacteria. Cyanobacteria photosynthesize, but not with chloroplasts. Because, of course, our cyanobacteria is a bacteria with no membrane-bound organelles, so they don't have chloroplasts. We also have autotrophs. Autotrophs obtain their food from eating other organisms. An example of that is E. coli. With that in mind, archaea and eubacteria do different things. Most archaea are chemoautotrophs. So remember, auto means self-feeding, and chemo is chemicals. So they feed themselves through the use of chemicals. There are three types. The first is methanogenic, or methanogens. These use CO2 and produce methane. I remember that because genic means produce, so they're producing methane. We also have methanotrophs, which means these methanotrophs are consuming methane and producing other waste products. So there's methanogenic and methanotrophs. We also have our second kind, sulfur reducing. So basically they use hydrogen sulfide in order to produce energy. And our third kind is nitrogen fixating. So they use nitrogen gas and they fixate it, which means basically they're having chemical reactions, and the end products are compounds that contain nitrogens. And these nitrogen fixating bacteria are really important for the nutrition of soil, because plants can use these nitrogen products made by these bacteria. You probably would have learned about this when you talked about the nitrogen cycle. Eubacteria, we have two kinds. One, heterotrophs, so they eat other bacteria. And two, photoautotrophs, so they can self-feed using light. But again, no chloroplasts. Now I wanted to talk about cyanobacteria. Cyanobacteria are a type of eubacteria, and they appear as blue-green algae. That's what they look like. Cyanobacteria are really, really cool because, well, they can perform photosynthesis. No chloroplasts, though. And they were likely the first photosynthetic organisms. So the first organisms ever to use sunlight to produce their own energy. Even more interesting, their ancient forms were likely the ancestors to chloroplasts. We're going to talk about that more in our next lesson. The last thing that makes cyanobacteria really interesting is that in the history of Earth, they are the bacteria that helped to oxygenate our atmosphere. So as I'm sure you remember from our discussion, class discussions about climate change, you would know that oxygen makes up about 20% of the atmosphere, nitrogen nearly 80%, and there's a few other gases in there, including things like carbon dioxide, water vapor, and other miscellaneous gases. But Earth's atmosphere didn't always look like that, or didn't always contain those levels. There used to be way more carbon dioxide and little to no oxygen. Cyanobacteria were really important in oxygenating our atmosphere, putting more oxygen into our atmosphere. Without them, things would look pretty different. Next up, we need to talk about metabolism. Now, metabolism in a general sense is basically just a term that describes the chemical reactions occurring in a living creature. There can be different types. You can have a metabolism that requires oxygen. So one that requires oxygen is called aerobic. So you need oxygen. And these would be considered obligate aerobes. So you need oxygen. You will die without it. Humans were obligate aerobes. We also have anaerobic. So anaerobic metabolism does not use oxygen. It uses other compounds. There's two types. We have obligate anaerobes, which means if there is oxygen, they will die. And we have facultative anaerobes. They can go with oxygen, they can go without. They're neither here nor there. Now, as you know, all living creatures need a home or a habitat. So eubacteria and archaea are no different. Eubacteria are just everywhere, literally everywhere. Just, you think of it, it's probably there. soil, water, organisms, it doesn't matter, everywhere. Archaea are known as extremophiles. I mentioned that earlier, which means they love extreme conditions. Philic or phile means to love. So they're extreme lovers. So glaciers, oceans, salt deposits, thermal vents, geysers, whatever, they love those extreme conditions that other organisms would not be happy in. So there are 4 types of extremophiles. We have thermophiles. Thermo means heat, so they love heat, an example of which could be a hot spring. Next up, we have the opposite of that. If thermo is heat-loving, we also have psychrophiles, which is cold-loving. There are also halophiles. Halo means salt, so they're salt-loving. So you would find them in the high salt areas like the Dead Sea or a salty lake. And we also have acidophiles, so acid-loving bacteria, which like to live in low pH waters. So eubacteria are everywhere. Archaea are our extremophiles in weird and wonderful habitats. We have a few images here. Up here we have a hot spring. You can see the steam coming off, and so you'd likely find a thermophile there. We also have thermal vents, and depending on the vent, they can be hot, so thermophiles. They can be salty, so halophiles, and there's often many gases that can be toxic to other organisms, so thermal vents, Archaea love them. And we also have this pink lake. There's a few of these in the world. I know there's one in Australia. And this is a very salty lake. So again, those will be our halophiles. Now, we started off this lesson talking about bacteria and how, they've got a bad rap. We consider them to be germs or bad. But the reality is they're a really important part of the ecosystem in so many different ways. They're decomposers, so they help recycle nutrients. They're nitrogen fixating, which allows plants to acquire nutrients that they need. They're also really important for our body. We have really important gut bacteria that help us to digest and acquire nutrients. Our skin is covered in bacteria. This bacteria can be positive, negative, or neutral. One positive sort of bacteria is where our natural microbial flora, so the bacteria living on us, could potentially compete for the space on our skin more effectively than bacteria that are coming from our surroundings. Of course, they do also cause diseases in other living organisms, and then humans also use them. I would encourage you to take a look at 5 of the coolest partnerships between animals and bacteria. It is a video that's kind of neat. And when bacteria interact with other organisms, there's a few different types of interactions that are important. The first is mutualism, an interaction where both parties benefit. So we keep talking about those nitrogen fixating bacteria. Well, the bacteria produce nitrogen that the plant uses, and the plant produces sugar that the bacteria can consume. So it's mutually beneficial. A similar thing occurs in our gut. There's also an interaction called parasitism, where one party benefits and the other is negatively affected. This is often the case when we have bacteria that cause disease or are pathogenic. Now, pathogenic bacteria can cause diseases, and we use antibiotics to treat these diseases. One huge problem in medicine today is called antibiotic resistance. What has happened over time is that due to overuse, patients not using them properly, or just by natural selection, many bacteria have become resistant to these antibiotics. The antibiotics kill bacteria or prevent their reproduction, and bacteria just aren't responding the same way as they used to. What has resulted is something called a superbug. A superbug is a strain of bacteria that are resistant to several different types of antibiotics, because we have a lot of antibiotics out there. This is, of course, a problem to human health, because if humans are getting sick from bacteria and we can't treat them because they're becoming more and more resistant, then, well, it's bad for the person who's sick because they're unlikely to get better. So here's a little image of that process. Basically, what's going on is we have bacteria hanging out, and we have a few bacteria in red that are resistant. Somehow they became resistant to an antibiotic. And then we are exposed to antibiotics, and that kills off most of the blue guys. But the resistant bacteria survive and reproduce, which creates more resistant bacteria. And this process can actually repeat itself with further exposure to antibiotics, resulting eventually in this superbug. So again, this concept of a superbug has severe implications on how hospitals and doctors can effectively treat patients. Okie dokie then, that is finally it. We have talked about the structure of bacteria and different components like the pili, the cell wall, the flagella. We've talked about gram staining, whether it's positive or negative, purple or pink. We've talked about shapes like bacillus, coccus, spirillus. We've talked about reproduction, whether it's binary fission or conjugation. What types of nutrients these organisms use and produce, and the roles of bacteria in our ecosystems. That's it for bacteria. As always, let me know if you have any questions, and good luck.